Hey, welcome to high school biology. My name is Rex. If you're meeting me for the first time, at the age of 13, I wrote my high school external examination and I cleared my papers. I'm 14, but I'm still too young to go to university, so I just keep up with self-study. I'm here with you right now to teach you and learn with you all I've learned in high school biology. Let's get to it. So before we start with biology, I'd like to define what biology is, the definition of biology. Biology is a branch of science that studies life. We all know that science studies all natural things around us, but biology is that aspect of science that studies life alone. There are two main branches of biology. There's botany and then there's zoology. Botany studies plants and zoology studies animals. The two other branches of science are physics and chemistry, just so you know. Biology is gotten from two Greek words, bios and logos, which means study of life. So biology is practically the study of life, like I said before. Next is the scientific method. I explained the scientific method in my previous video, Introduction to Chemistry, but I'm going to explain it here and I'm going to give you a scenario of what I'm talking about. The scientific method is a series of steps taken by scientists to make breakthroughs in their field. In biology, the scientific method helps biologists to make breakthroughs in their field, which is biology. I'm going to give you a scenario, like I said, to explain these series of steps. First of all, I will list them out. There's an observation. Then there's identification of problem. I'll just put ID of problem. Then there's a hypothesis. Then there's an experiment. And then there's a conclusion. Then there's a theory, and a law. Note that when you make a hypothesis and you want to conduct an experiment and your experiment doesn't fit with your hypothesis, you can always go back, all right, to that hypothesis. The good thing about a hypothesis is that it can easily be dropped and replaced by another hypothesis. So, like I said, I'm going to use a scenario to explain these series of steps. Picture this. It's 1928 and Sir Alexander Fleming had discovered penicillin, that wonder drug that cures bacteria. But how did he do it? He was studying the growth of a particular type of bacteria called Staphylococcus. And he was using agar to study them. Agar is a powder that the bacteria can easily grow upon, and you can easily see the bacteria or any other bacteria or mold that is in the agar. It's like a powder. And then there was this mold that had gotten into the petri dish that he was studying the bacteria with the agar by accident. And this mold was called Penicillium notatum. And he noticed that where the penicillium was growing, the bacteria around there had died, creating a clear zone around the penicillium. He had made an observation, and then he identified the problem. Now he was asking himself, what had caused this bacteria to die? Like, what was in the penicillium, or what was in the penicillium that caused the bacteria to die? Well, he proposed the hypothesis that there was probably a chemical in this Penicillium notatum that diffused into the agar and killed the bacteria. And so he designed and conducted his experiment. Since he was using nutrients to feed the bacteria and the mold was growing around the bacteria, he used nutrient broth and put the mold in nutrient broth so that the chemicals from the mold would diffuse into the nutrient broth if his hypothesis was correct. And then he put the nutrient broth minus the mold because the chemicals from the mold had already diffused into the nutrient broth, and he put it into the petri dish where the bacteria was growing, and all the bacteria died. So at this point, you might be thinking, okay, we're going somewhere, but he decided to try out something else. He wanted to know if the mold wasn't added into the nutrient broth, would the bacteria still die? If the bacteria died, it means that his hypothesis wasn't correct. So he made another nutrient broth, but didn't add the mold, and put the nutrient broth in the petri dish, and the bacteria did not die. This means that his hypothesis was correct, that it was the Penicillium notatum that was killing bacteria. This was the creation of penicillin. As you can see, I used the scenario to explain the observation, identification of the problem, hypothesis, experiment, conclusion, and after so many times, after so many consistent results of the same experiment and giving you the same solid results, he ended up with a theory and eventually a law, which was the creation of the antibiotic we know as penicillin. Note that if Alexander Fleming's experiment didn't work according to his hypothesis, he could have gone back to postulate another hypothesis and then conduct another experiment. Now that we've dealt with the scientific method, I'd like to talk about some of the usefulness and applications of biology. I'll talk about the applications first. Where biological research has really helped us in the world are mostly in vaccinations and creation of new medicines for curing diseases and preventing them. Also, the use of in vitro fertilization, or IVF, to help infertile couples to have children, and the use of hybridoma technology for antibiotics. It's okay if you don't understand what hybridoma means. We're going to study that as we study biology more. If you're interested in the biological field like I am, there are several options to choose from. I'll give you a few, but I advise that you do some research of your own. There's the medical, veterinarian, and even dentistry industry. There's the agricultural and horticulture industry, and that even that hybridoma technology I was talking about is among biological applications. Now that we've dealt with the usefulness of biology, I'm going to talk about the characteristics of living organisms. How can you tell a living organism from a non-living thing? If I bring a dog and a ball and I ask you what is the difference, you will be like, oh, the dog grows and the dog eats, the dog excretes, the dog respires, the dog can move from one place to another, and the dog can even respond to its environment. But no matter what you do to a ball, kick it, bat it, throw it, toss it, it's still going to remain the same. The ball doesn't grow, neither does the ball start to breathe, neither does the ball start to excrete or even eat or, gain nutrients. So what is truly the difference between living organisms and non-living things? That is what we call the characteristics of living things. I use this acronym called Mrs. NERD to help me remember the characteristics of living organisms. So here they are. There is nutrition, there's movement, there's sensitivity, there's excretion, there's respiration and reproduction, and then there's growth. The Mrs. NERD, so M is movement, R is respiration, S is sensitivity, N is nutrition, E is excretion, R is reproduction, and G is growth. Note that a living organism cannot be called one if it doesn't follow any of these characteristics or even one of them. And note, this doesn't only happen with animals. This also happens with plants. Now that I've talked about the characteristics of living organisms, and I've also shown you that those characteristics apply to both animals and plants, I'm going to talk about the difference of characteristics between plants and animals. So I'm going to write down that acronym I told you of earlier, which is Mrs. NERD. Ooh, spill of ink. NERD. Okay, so the M stands for movement. R stands for respiration. S stands for sensitivity. N stands for nutrition. E stands for excretion. R stands for reproduction. And G stands for growth. Sorry, let me clean up this mess here. These 7 characteristics of living organisms, movement, respiration, sensitivity, nutrition, excretion, reproduction, and growth, apply in different ways with plants and animals. I'm going to talk about movement first. Plants don't move the same way animals move. Plants don't really need to find anything, so they are fixed in their roots, since photosynthesis only occurs from the sunlight and in their roots, so they don't really have to move much. And you know that photosynthesis is a way that plants make their own food. But animals need to move to find food. This difference in movement, or in nutrition, I would say, is autotrophic and heterotrophic nutrition. So I'm extending both the movement and nutrition together. Because, sorry, this ink problem. Because animals move to find their food, and plants don't move because they already have their food in them. So this characteristic in plants is called autotrophic nutrition. Sorry, autotrophic. And this means that plants are autotrophs, but animals are heterotrophs, or they have heterotrophic nutrition. Heterotrophic nutrition. All right, I've talked about movement. Now let's talk about respiration. Due to photosynthesis, plants respire. They don't really have any special organs for respiration, and they respire through the air. So that's the difference between plants and animals. Animals have specialized organs for respiration, such as the lungs, gills, and trachea. Respiration is done. Now let's talk about sensitivity. If I were to sneak upon you and suddenly tickle you, what would you do? Or how would you react? You would suddenly jump up, right? That is sensitivity to someone's environment, or sensitivity to stimuli, which is the hormone of laughter. Okay? But so plants don't respond quickly to stimuli. Anytime something happens, they don't really respond quickly because they're in a fixed position. But animals will respond quickly because they have different organs for movement. So if prey gets scared because a predator is coming, the prey receives a stimuli or hormone of fear and runs or flees because it has organs for movement, like I talked about earlier. I talked about sensitivity. Now, nutrition. I already talked about nutrition when I said that plants and animals don't make their food the same way. Plants make their food themselves due to photosynthesis, but animals have to find their own food, either eating plants directly like herbivores or eating animals that eat plants. So what I mean is there are different types of heterotrophs. There are the herbivores, carnivores, and omnivores. Herbivores eat plants, carnivores eat animals, and omnivores eat both. So basically, animals eat plants directly and indirectly, either as a herbivore, which is directly, or indirectly when an animal eats another animal that has eaten a plant, or has eaten another animal that has eaten a plant. Okay, so this is heterotrophic nutrition, and that one of plants making their food is autotrophic nutrition. Next is excretion. Plants don't excrete the same way animals do. Plants don't make a lot of waste products because the process of photosynthesis is very simple. So they release oxygen as one of their waste products, and any other waste products will be stored within them because they're not harmful substances. But animals engage in a lot of activities, and they have a lot of waste within them that they have to pass out. And there are several organs for that. Some examples are the kidneys and the Malpighian tubes. So that is it for excretion. When it comes to reproduction, animals have specialized organs, or gametes, for reproduction and fertilization. The sperm for the male and the eggs for the female. Plants reproduce in an entirely different way, and I'm going to explain that to you as we dive deeper into our next topic, classification of living organisms. So stay tuned to find out. Reproduction is done. And now for growth, there are two things I want to talk about. Plants have no symmetrical growth. It means they grow with no definite shape, okay? And they can grow outwards and keep on growing. But plants have a fixed symmetrical shape, and once your growth is done, it is done. It doesn't, they don't grow for infinity. And the last part I want to talk about, which wasn't part of the, characteristics of living organisms, was the way their cells are structured. Plant cells are different from animal cells. Plant cells have a cell wall in their cells, while animal cells don't. So, plant cells have that rigid structure because of the rigid cell wall. Therefore, animals need to have a protection, which is either an endoskeleton or an exoskeleton. Exoskeleton are those skeletons that are outside, like beetles or butterflies. And then endoskeletons are those skeletons that are inside, like us. Here are the questions. Number 1. If you're interested in the field of biology, what would you like to be as a biologist? Let me know in the comments. And number 2, give me a difference between plants and animals when it comes to the characteristics of living organisms. Among the ones I mentioned, you can pick any one. It's fine. If you enjoyed this video, please like, subscribe, and share with at least 3 people you know don't have access to basic education or who's bold enough to explore homeschool. And I'll see you in the next class.